the Prion-like spreading hypothesis, it says that alpha-synuclein, which is the protein in cause in synucleinopathies like isolated RBD or Parkinson's disease, it says that the alpha-synuclein becomes misfolded and it starts spreading from one cell to the other. And this is why someone has a propagation and the manifestations that we know are associated with Parkinson's disease. So according to the Prion-like hypothesis, it's going to spread from one cell to the other. The other hypothesis is the selective vulnerability hypothesis, which says that it's not really that it spreads from one cell to the other, but it's rather that some peculiarities, some particularities in the cells make alpha-synuclein more at risk of becoming misfolded. So it's not really that it spreads from one cell to the other, but it's something that happens like in a cell for particular reasons, vulnerability reasons due to morphology or whatever. So what we did in our last paper is we developed a computational model that showed that we can actually recreate the atrophy that we see in isolated RBD by propagating computationally in silico on the computer the propagation of a misfolded protein through the brain. And this model simulates the spread based on connectivity between brain regions and based also on the selective vulnerability of a region. And we showed that both factors are important. So if you want to recreate the atrophy that you see in RBD, you need the connectivity of the brain. So it has to go through the actual connectome. And you also need to take into consideration the selective vulnerability features of the brain region in question. In our case, it was genetic vulnerability. So the more SNCA was expressed in the region, or the more GBA was expressed in the region, the more vulnerable the region was to developing atrophy. And this corresponded to the actual atrophy that we saw in patients with isolated RBD. So in terms of your question, both connectivity and gene expression, so both Prion-like features and selective vulnerability features are determinants of the spread of alpha-synuclein in the brain based on our understanding of atrophy.